Okay, well first, thanks very much for the organizers and for everyone else. And to me at least, the fact that there was a lot of arguing here meant that there was an awful lot to argue about. It was interesting to argue about. I think that's somehow the crucial part. Okay, so I just wanted to make a few comments, none of them very profound, but I think they're all things that are worth comments that are worth making. And this is from the perspective of a condensed matter physicist, and if anyone's interested, I can explain why condensed matter is a relevant adjective there. So I'm just going to say a few comments first about numbers and general issues, some vague things about evolvability, and trying to tie a bit of things together maybe, and then a bit about models and extrapolation from those. Okay. So the first thing is how big are the numbers? So the time on Earth, which is what's always emphasized by anyone talking about evolution, is a very long time. Even for bacteria, the number of generations is substantially less than 10 to 12, usually much less. That's sort of the upper bound. But the total number of bacteria on Earth is really enormous, estimates in the 10 to the 30th range. Right? Which means if you ask, what are the total number of bacterial cell divisions ever? So the number of experiments done by evolution, or the number of tenths at finding something better, whatever that means, that's the product of these, which I'm calling A for convenience. And that's numbers of order 10 to the 40. And most of that number comes from the very big population. It doesn't come from the time. Right? So if you want to emphasize big numbers, the number to emphasize is the number of individuals, which is to do with the size of the Earth. If you say, okay, bacteria are boring, you're interested in vertebrates, then the total time of generations is less than 10 to the 9, that ballpark. But the number at any given time is bigger than 10 to the 13. And so again, the big number, which is the product, is dominated by the total number, and it's very large. I mean, it's ballpark Avogadro's number, possibly. Okay. There are an awful lot of other numbers, and these we've no idea how to even define them. Sort of number of niches, which in a very loose sense is the number of possible problems to solve, and that's one that's come up various times here, and is an extremely important part. Is that it doesn't try to solve one problem, it doesn't try, but it ends up solving one of very many. That's of course associated with complexity of environments and organisms. You know, how one can characterize these by dimensions and things came up in Carl's Wilcham's talk there. You know, fitness is obviously something which is plural, it occurs on multiple time scales and so on. And there's all kinds of things which come in here, which there are numbers that we know, like sizes of genomes, but you know, why they're the sizes they are or the significance of that, we've no idea. Okay? And of course, rates of genetic changes and the processes there, which ones dominate, particularly roles of recombination and gene transfer that various people have also talked about. Okay. So polynomial or not? Okay, so this is a gang of 5, maybe more than 5. I think 5 were involved in talking. So just to avoid confusing notation, so I'm calling K something like number of variables involved. I think it was called little n. The number of mutations needed to cross some fitness valley, or some generic term like that, which is somewhat how complex it is. So if I have a problem, if it's non-polynomial, if it's hard to solve, but it happens to be that the number of attempts needed goes as e to the k to the 1/3, so the complexity goes as e to the k to 1/3, so it's not polynomial, but it's not a simple exponential either, then, you know, if C is 1, you can get k of 10 to the 6th on evolutionary time scales. If C is 8, which if I plugged in a stupid estimate from what I learned the other day from factorization, then k is of order 2,000. So k is what you would call little n.
So
yeah.
To get to x for 100.
Yeah, so to get to e to the 100, right. I can put in 1,000,000 here and I get e to the 100. Okay? If I just plugged in for k to the 1/3 for factorization, and I needed a C of 8 to fit this to 125 roughly, and then the ballpark is, okay, you can get to a couple thousand. Okay? So it's very big numbers because the basic number of attempts possible is extremely large.
So your point is that there is one bacterium somewhere that knows how to factor
no.
a million bit number.
The point is that I really have to look at the difference. I can't just say something's non-polynomial, therefore it's really hard. It's hard, but one has a hell of a lot of attempts that evolution has made. Right? So I'm using this e to the k to the 1/3. It comes up in other contexts, finding spin glass ground states, and so on. And it's sort of a borderline case, and I'll come back to that briefly. Okay. The other one is that if it's polynomial, so if processes are polynomial, and so they're going as the k to some power, then a really interesting question is, does recombination change that power? Right? It's still polynomial, but maybe it's a faster polynomial, an easier polynomial with recombination or with some other processes, and that's a really important one too. Or does it depend upon a lot on the selection regime and how the process is carried out?
And here Nick's talk, Nick Pippenger's talk, suggests that you can go from at least 10 to n to square root.
Yeah, so that's a concrete result in a particular context, actually I'm going to mention in a minute. of where one can say how things depend on some of the parameters, and that's really asymptotics in various regimes. And I think those kind of results are very instructive. Sometimes one can apply them directly to experiments, and that's of course at least something one can say concretely and make contact with the real world. So I think a very important point which Paul Bellion emphasized, what should one be surprised by? And one of the big advantages, to me at least, of trying to show which things may seem difficult, but in fact are polynomial evolvable, means, okay, those ones we can ignore. We don't have to be surprised by those, and we can try to pin things down on the ones that maybe we do have to be surprised by still. And maybe those are extremely rare. Maybe evolution, to get to where it has on Earth and the diversity and so on, needs rare events with probability 10 to the minus 35 or 10 to the minus 40. Okay? Or maybe it doesn't. We don't know. We don't know which things really need rare events, which ones don't. And this is the question, does the size of the Earth matter? Right? If I don't need such rare events, and I make an Earth 10 to the 6 times smaller, but with similar diversity of environments in it, then will evolution be different, or will it be roughly the same? Right? And so that's the question of whether the big numbers really matter, or is it only time? Sorry?
What do you do for an atmosphere, 10 to the 6 smaller?
No, I can constrain, keep all the geophysical parameters the same, right? But just change the size. Okay, now in practice, I may not be able to do that with existing physical laws. I probably can, but it's not. There's so many possible planets around we're discovering it. I don't do it to the atmosphere. This is this is God's or some other God's role than the one that we've had so far. Okay, so one of the things which isn't also an important observation, and this is true on short time scales and long time scales, is if rare events are important, one can learn something about asking which are the rarest events that are still ones that can occur in the time scale and population size of the experiment. And if there's one which is less unlikely than all the others but can still occur, that may be the one we see, and so there's some predictability about those kind of rare events, right, even if they're extremely rare. And there are simple situations, at least, in which one can understand some things from that. Whether you can understand in any big picture, complicated situations, I have no idea. I certainly don't know how to. Another important thing that's come up is sort of the nature of the selection, and a very crucial thing here right from the beginning is this combination I wrote down, the total number of evolution experiments ever carried out. Is that the right number? Or is it some other combinations of times and population sizes? Does it depend hugely on how I structure my population? Is it a well-mixed population, or are they competing in some more subtle way, particularly in Worm's talk? He emphasized things that you can get a lot more diversity in a population. I think everyone will agree that diversity is crucial. You can keep a lot more diversity in the population and avoid things which are pretty good but not that good to take over if you do the selection differently, if you have things mixed and so on. And an obvious way that happens is spatial structure in the real world, and whether the competition is local or global, how things mix back together and so on, and the enormous range of time scales that come in even to that. Okay. Then a much more important one, which I'm not even putting a question mark after this, I think this is absolutely crucial, is time-dependent environments. There is not a fixed set of things that could be done, fixed fitness. It's changing in time. It's changing on multiple time scales. And as Watson talked about, and Kusel talked about some of these things as well yesterday, and that's really crucial. If one understands things things that are easy in a fixed environment is great, then we don't have to worry about those. But there are certainly a lot of things that can happen when I have time-dependent environments that may not be able to happen in fixed one. And I think for understanding anything to do with reality, that's absolutely crucial. One of the specific contexts in which the creation and maintenance of diversity came up was in the immunology context in a couple of the talks. And the nice thing about immunology is, even though it's awfully complicated, it's very small bits of DNA that it depends on, both in the case of the virus and in the case of the of our immune system. And so it's one could hope to make progress on. It's also really fast. So if one wants to get quantitative understanding. So another thing, and this Ray Christoph mentioned, once one starts saying we don't know what the combination parameters are, we really have to talk about different regimes, and we can ask in specific contexts at least how long the time to evolve something is. Does it go with 1 over population size, as would be suggested from this combination here, or 1 over square root of n? Daniel Weissman gave an example of that. And in Pippenger's talk, then, does it go as the number of alleles you're trying to get, which here's k, or square root of that? Well, and that also makes a big difference, right? These are numbers, and which ones occur make a big difference. And one really likes to understand regimes, what works in different regimes. And any of these results have some amount of, in a loose sense, of universality to them. Do they apply in context other than the specific model you calculated with? right? And that's always the bugaboo here. You do some model, you leave out almost everything, and the question is, have you captured the essence of something? And so you have some hope of generalizing it, at least as the kinds of behaviors, right? And so you can try to extrapolate and things like that. So the evolvability paradox, I mean, I think it's probably enough discussion of this that maybe don't need this, but I would like to show this in a talk with physicists and some others. Because of there is this, in some sense, paradoxical aspect of it. So I have some measured property here, and I'm measuring this as a function of time, okay? But I'm not going to tell you which way time is. Now, if you're a physicist, you would look at this and say, okay, I'm coming down into some valley, I'm approaching that, and then I'm fluctuating around and going like this. Obviously, time's this direction. If you were an evolutionary biologist, you'd say I'm climbing up to a fitness maximum, and I'm sitting near there and something like this, okay? But I didn't tell you how I did this plot. What I did was I drew a bar right here, I drew a bar there, time went this way, time's going up, and then I just waited until the first time it crossed here. And then I stopped and I showed you what it looks like. Okay? It turns out that looks exactly like, for simple context, as it would if I turned it backwards, the typical one. So I'm looking at a very rare event, but I'm looking at a rare event that reached some threshold. Okay, what's that threshold? That threshold is where the organism is successful enough to get in the fossil record or to have existing organisms descendants now. Right? So everything is conditioning upon things being extremely successful in the future. And anything that does that, very generally, will have some aspects to it. This looks as if it's systematically climbing up this hill. In fact, you can say, well, law, this looks as if it's following when it climbs this hill. Okay? Or goes into the valley if you're doing it upside down with fitness. And so you can say some things about it, but the properties of it are really dependent upon this conditioning upon something very rare happening. Okay, and that's really crucial when one tries to talk about things like mechanisms. Always conditional upon long-term survival, and that's anything like that is always going to appear as if it has some features of being evolvable. Okay? This is something where the time scale of fitness is absolutely crucial. This is on long time scales, which we require for things we actually see. Okay? In the context of biology, there are various mechanisms associated with this. They're apparent in some sense, but they're of course real mechanisms that did evolve. Okay? But they're apparent in that they evolved for something. And various speakers talked about these. The computer science use, I guess, is a somewhat different sense of evolvability we've heard here, but what was focused on is a solution of one hard problem. And Paul talked about the context of guessing a password. Okay? But, you know, if you're a virus, you don't have to guess anything about the immune system of a particular person or even a particular species. All you do, you've got an enormous number of species, enormous number of people you could attack. And if you get into one of them, that's fine. Right? There are a lot of you can try one of those. So there are a huge number of possible problems. And this is a really crucial part, and I guess came up in one's talk of things are much easier if you've got a lot of possible problems to solve, or many possible solutions in that case. And this came up in sort of a looser way in several people's talks that I'm going to mention again in a minute. Biology finds its own problems to solve. One of the absolute classic things that comes in all over the place, if you try to do evolution experiments, you try to select for something, the biology chooses something else. Okay? And that's always true, and there's a nice story about oral exams on how you use a barometer to measure the height of a building from physics, which I won't say given the time. But it's a rather good analogy. Believe me. Someone can ask at the end, and then I can say, okay. So another sort of general thing which has somewhat come up, but I think rather less so, is how much sort of evolvability and biology and everything generally is associated with sort of being on a borderline. Okay, and I think the thing is the only person I'll quote, and he's alive. I don't like quoting dead white males, live ones, okay, maybe occasionally. The term "the edge of chaos" I think is Stuart Kaufman. So organismic biology, there's one view of it that everything's very modular. And everything does this, does that. But that is a problem. It's very hard to, everything that's modular, how do you get it to do something new? That's very hard, or even really to change a lot what it's doing. On the other hand, it means it's robust if you change other things and so on. The other one, you see these protein connection networks, which are nonsense. But those, they look like random networks, and there are some aspects of actually the functional parts of the biology that also look sort of like random networks. But of course, if it's a completely random network, it probably can't really do anything. And so probably in some very loose sense, biology hovers on the middle. The border between these, the biology that's been evolutionarily successful. Not all biology that went extinct a long time ago. Okay. Another one like that, and this has come up, is whether selection is on individual genes or selection is on the whole genome. Right? Can I really think of selecting on this gene or selecting on that gene? Or are there enough interactions between genes that I have to think of selecting really on the whole genome? Okay, which is an straight asexual evolution. You select on the whole genome. Now, but of course recombination breaks things up, puts them back together again. So there's a real suggestion that, sort of natural and probably in some real sense optimal is for the organisms to be on a borderline between selection on genes and selection on genomes. Particularly Schramm and Nair have thought about that recently. I think that's a very interesting general issue. Okay. Another sort of borderline one is on timescales. There's timescales usually one thinks of for generating a variation, that's sort of slow, at least for new mutations, and the selection, which is relatively fast. But then really all these occur on all different timescales, depending on the processes, and this came up in Watson's talk. But probably having these on the same timescale, at least some of the time, is probably important. So that's another one, maybe sort of on the borderline between two different things. In the point of view of the complexity, and the reason why I put down this example, is one might think, given the borderline between having so many tasks you could possibly do, but none of them are of much interest, and not being able to do anything, there is a borderline where you can do, there's a lot, but you can do some of them which are sort of interesting. And if one side of that thing is polynomial...
There is another function there, this what we solved. I don't know if you like it better.
E to the log squared, I love.
X to the square, yes.
Yeah, I love e to the log squared. Yeah, that's really fast. Right? And that comes up, there's a context, actually, Oscar Halacek, where that comes up, those e to the log squared kind of things. But if you're then on sort of the borderline between here, you might say, okay, I'm on the borderline between a polynomial and exponential regime, and there's some kind of behavior in the marginal behavior there. And I believe that happens in random cases in at least some of KSAT on the borderline, right on the borderline, is at least seems that the algorithms are something which are between polynomial and exponential, but I'm sure someone can correct me on that. For random cases. So this sort of idea that you're somewhat on the borderline seems to, comes up in a lot of contexts, and of course, the evolution doesn't stick on the borderline, it'll go off one way or other, but if it goes off one way or the other, its lineage will die out. And so you're conditioning upon, in this case, loosely speaking, only one parameter, which is that the things you see very long-term survival sort of stay around on this borderline, and that's a sort of loose one-parameter sense of evolvability. So the last thing I just wanted to say is going away from the general to the sort of specific models. And this is something which, as a theoretical physicist, one tends to often work on specific models, and then you try to scratch your head and say, wait, is there something about this model that's general? Can I go beyond that? I want to solve it not exactly, but by sort of asymptotics and understand, so one gets the answers right, even if one can't prove them, or at least one has something that's either definitely right or wrong. And then one tries to go from there, instead of so much thinking about classes of problems from the beginning. Right? So the physics is more like you start with one problem, and then you try to build up and say, okay, there's sort of a class around this. Which is maybe easier in some ways than some of the computer science approaches that we've heard about. Okay, so one example of this was the prebiotic autocatalytic sets that Mike Steele talked about, and he didn't really talk about how you make different scales of that model, but that was sort of hinted in there. And it's a toy model, but it's something that sort of has a smell of it, of something like actual biology. Then there's morphological development that Eric Sidja talked about, which also has a smell of biology with a much further stage along in a morphological development. And then there's lots of toy models that Watson talked about, which are fascinating, and I think one is really likely to be able to learn a lot from, but they're still more the character of toy models. We're not trying to put too much biology in them, because then one can't explore as much around, but they sort of have some bits of them. I think Francis Crick calls them just so models. Of you make up a story for something, but this is not done in, just in words, but it's really done with model. And there's a huge amount one can learn from them, and one may realize, wait, actually some of those are actually closer to more realistic things as well. Now the difficulty of all of these is extrapolating, if you think you're doing something connected to reality, to real numbers, meaning to big numbers typically. If one wants to make things more formal, from some of these one would like to define some sort of class of problems of more difficulty. Right? It could be involving more complicated patterns in Eric Sidja's, or bigger autocatalytic sets with other properties in Mike Steele's, or various things that I think come up naturally in Richard Watson's. But you might want to find some measures of the size of the problem. Again, I'll call that K, or like a little n. But then I have to, of course, immediately say, what are the number of possible problems that could be solved to do well? Right? There isn't just be one problem, about there are many, and that was already explicit in, I think, all of these three examples that we heard about. Again, I already mentioned the one with the virus, right? There's many possible hosts, even something simple as a virus. And then one would like to get, the computer science question about how difficult is that? that difficult to do this as a function of k, and if one finds it's e to the k, okay, well, it's not going to do too much. If it's some borderline case, then maybe one still can, but that one should be moderately surprised. And then, of course, it's going to depend upon the nature of the competition, the recombination processes, and so on. So we'd like to get sort of concrete results on those aspects.
I might be somewhat misunderstanding the possible problems to solve, but isn't it true that if you have more hard problems to solve, that's actually not a good thing?
So it's not a good thing for solving any one of them. Yeah. But if I have many hard problems, and all I need to do is solve one of them, right? It doesn't matter which one I solve one of them. So if it's exponentially hard to solve any one, but there are exponentially many that are fine if I solve those, then at least naively those exponentials can balance each other and it can become easy. Okay, now obviously that's a very loose statement, and there are classes of things that one can talk about and try to make sense of it. And I think that maybe are some natural classes that might come from some of these or from some simpler models. And particular sort of I think in Richard Watson's, one of his of having different patterns, well, you can start going, well, there are many more patterns which you can do well by getting something like those. So evolving to remember 10 patterns may be very hard, but if it's any 10 of a million, already that might be somewhat less hard, right? So I think those kind of things are a really important one to get. And then, of course, one would like to go back and ask about questions about any kind of universality. Is there anything that one is learning which is from some models, which in the limit of some of the numbers being large, right? So lots of large, lots of small numbers, that's the kind of regime where universality can often hold, central limit theorem being the most famous example. Is there some kind of universality in these large number limits of something one can say beyond the very specific model? Okay. I just want to just end on one note of actually coupling to an experimentally observable statement. Well, not experimentally observable, but observed in genomic history. And that's a question of, do we have some examples of where there seems to be some sort of well-defined limit to complexity? Okay, and I don't know if this is one, but it seems at least one is useful to think about. Particularly if one is thinking that one of the things one is going to look at is gene regulation and trying to understand gene regulation. And bacteria are very simple here. They have conveniently a factor of 100 range of genome sizes, so you can ask somehow how things scale with genome sizes. The smallest ones have essentially no transcription factors at all. They don't do gene regulation, the proteins do everything. Okay? The largest ones have a lot of gene regulation. And there's a very strong trend, and this is not just some R value, this is a plot which I should have pulled up, with remarkable scatter around it, that the number of transcription factors goes as the square of the size of the genome. Okay? And if you start putting in numbers, you realize by the time you get to the biggest genomes of somewhat more than 10 to the 7, the number of transcription factors is so large it's getting to be a substantial fraction of the genome. And it seems if you want to regulate anything that can do more things, more chemistry typically, than the bacteria that we have, and a bacteria that was 10 times as big, you would need more transcription factors if you had room in the genome. Okay? So somehow this kind of architecture of how the bacteria do things seems to have some intrinsic limit to it. Eukaryotes just do it differently. They have a major difference in how they do it, and they sort of random combinatoric methods. And I think understanding how that might have evolved is one of the most interesting directions. So here's one where one might actually try to...
Where does the G square is
observational. Empirical, yeah.
Yes.
So typically as bacteria get more complex, bigger genomes, they do more various things, but not by very much. They typically do much more metabolism. They can eat more different things, they can make more different things, they can do other stuff like that. And so you have to kind of regulate all these metabolic things, regulate the conditions under which you can live, so you're still happy in those and so on. And so somehow what you need to do gets more complex as well, and empirically the number of transcription factors grows quadratically, and so you basically run out of steam. Okay? And even microbial eukaryotes with similar sized genomes, they do it differently. And that grows much more slowly with genome size, the amount of transcription. So it's just an empirical thing of something one might say, okay, there is somehow some limit there. Okay, well, I'll stop there.